Good afternoon. My name is Sarah Dugan. I'm a mental health pharmacist and faculty here at Neomed. Our topic for today's didactic is the gut microbiome and inflammation. Our objectives for today are fourfold. So to define what the gut microbiome is, and there's a little bit of discrepancy in terms of terminology. Microbiota is what is sometimes referred to when we talk about the relationship between humans and all of the microorganisms that make up the microbiome. The microbiome sometimes being referred to only when we're looking at the microorganisms and their genes themselves. They have been used interchangeably. For the sake of ease in this particular presentation, I will be using microbiome. Sometimes we're referring just to the microorganisms, sometimes we're referring to humans, sometimes we're referring to both, but that will be the terminology I use today. We're going to list some common organisms that have been identified in healthy humans. So that symbiotic relationship between some of the microorganisms in the microbiome and the human species. We're going to identify some correlations between the gut microbiome and medical conditions. I just want to point out we're identifying correlations. So where a relationship has been identified, we are not identifying causation. So in a number of instances, you'll see that there are changes, but it's not necessarily which came first, that the changes in the microbiome drove the medical condition. It may very well be in the opposite direction, but just that there is a relationship there that's been noted. And we're going to describe the relationship between the gut microbiome and inflammation. We're going to look at two specific examples to help highlight this. So to start off, what is the gut microbiome? The microbiome is a collection of microorganisms. There was interest in the microbiome as early as the 1600s. There were some researchers back then that were able to obtain samples from within the same individual and compare that the microorganisms, mainly the bacteria they could grow back then, look different depending on where the sample was obtained from. So they wanted to proceed. Not only could the bacterial samples look different in a microbiome of a person from different locations, but as they started to compare those samples between individuals, they also started seeing some differences, including pretty significant differences between individuals who were healthy as well as those that were ill with a variety of different illnesses. The original interest may have been mainly with bacteria because that was something that they could grow and culture and study, but we've come to realize that it's beyond just the bacteria. It includes fungi, protozoa, viruses, a number of other prokaryote single-cell organisms that live throughout the body. Now, there are microbiomes that exist throughout the body. Interestingly, I came across the study that said that they are able to identify which finger you use to type on a keyboard, because your fingers have unique microbiome fingerprints, essentially. So the colonies of microorganisms that you leave when you push down the keys on a keyboard, they'd be able to tell you which of your fingers actually hit on those keys, which I don't think my husband would like, because he definitely just uses the 2 and then his thumb for the space bar, but that's about it. But they are able to differentiate that. For the sake of today's lecture, we're just going to focus on the gut microbiome. So looking a little bit more in depth at what the gut microbiome is, it includes a whole host of single-celled microorganisms as well as some simple multi-cell organisms. The organisms in the microbiome outnumber the human cells by a factor of about 10. So 10-fold more of these microorganisms than what we have in terms of human cells. When we look genetically, it's an even greater discrepancy. So there have been approximately 22,000 different human genes identified. So different versions of the genes have been identified. For the microbiomes, it's over 3 million, so estimated a little over 3.3 million non-redundant genes that have been identified through the microbiome. So recognizing that this microbiome, which sometimes is referred to as an organ in and of itself, may contribute quite a bit to an individual's functioning. That additional genetic makeup can help where humans aren't able to do something. It could help fill that gap. And that's where we get the symbiotic relationship. Hopefully, we're going to kind of look at how that may go astray sometimes as well. There are more than 50 different bacterial phyla that have been detected. We have some that are identified more commonly than others. Bacteroidetes and the Firmicutes are two of the more common strains that have been identified. And we're going to talk about some changes related to them in just a few slides, as that seems to potentially play a role with some of the inflammation that we see. So how does this start? Colonization of the microbiome is believed to begin at birth. Originally, if an infant is delivered via vaginal birth, their microbiome reflects pretty closely the vaginal microbiome of the mother. If an infant is delivered via cesarean section, their microbiome tends to represent more of a dermatologic microbiome of their mother. There are differences that are maintained within that first month of life for both of the infants, but by the time they get to month 6, they don't see as much of a difference between the microbiomes of the infants, regardless of the way that they were delivered. There are a number of parts of life that seem to significantly impact what the microbiome looks like. So birth being one of them, in the introduction of either breast milk or of formula. The first onset of a fever seems to create some changes in the microbiome. The introduction of simple foods, things like rice cereal, also tends to start to shift what the microbiome looks like. When an individual has table food introduced, again, they see a shift in what the composition of the microbiome looks like, and then the utilization of antibiotics. So those are different timeframes in most individuals' lives where we do see significant shifts within the individual in their makeup of their microbiome. There are a number of other both internal and external factors. External factors could be what the microbial load of their immediate environment looks like, the type of food that they eat, what their feeding habits are, the way that they eat, as well as temperature. Some of the internal factors that can play a role in what the makeup of the microbiome is include intestinal pH, other microbial interactions, so if they're getting them from some other source, environmental temperature, peristalsis, the makeup of their bile acid, immune response, stress, or trauma that they may be exposed to, as well as drug therapy. So this microbiome really can be influenced pretty significantly by both internal and external factors. So what does it do? So it could be influenced by all of these factors. I said there's a symbiotic relationship. So what can it do? Well, the microorganisms were originally really focused on their role related to digestion and nutrition. These microorganisms can live off of some of the non-absorbable carbohydrates, starches that are ingested by the human host, by the individual that they're living in. So they're actually able to take some of the food material that our body wasn't able to break down, and to break that down even further to create usable energy for our body to be able to absorb and utilize. So they really can assist us with that in terms of nutrition and digestion. They can form a barrier to pathogens. So along the lumen of the GI tract, there are a number of locations where there can be adhesion of different microorganisms. By having more protective or symbiotic organisms that line that, ones that are doing us a favor by breaking down food or doing other things, it allows less of an opportunity for pathogenic bacteria, for example, to get a hold and start wreaking havoc there. So they actually can help serve as a barrier. In some cases, they also secrete substances or chemicals that will kill off other microorganisms that may be pathogenic to the human host. They are involved in immune system signaling. There's a large number of them that are there. We talked about the number of genes that are there. Some of those genes actually can translate into either lipopolysaccharides or different molecules within the microorganism itself that may assist with providing a signal to the immune system that, hey, there's something going on here, or they may create and release other chemicals that may trigger the immune system when it's necessary, or sometimes we'll look at a couple examples where maybe they do it and it's a little extra sensitive. So we're getting something like an allergic reaction or an intolerance in some cases. Also of significant interest is central nervous system signaling. So there are signals, some of the chemicals that these microorganisms can create are able to go back and provide feedback to the central nervous system, especially the brain. And this has been shown to have a pretty significant impact in some individuals, those with mood disorders, with anxiety disorders, some individuals with post-traumatic stress disorder. There seems to be a relatively large role played by the signaling that's happening by the microbiome. And they see some changes within the microbiome for some individuals who are suffering with symptoms that really haven't responded to any other type of treatment. So it's involved in signaling in other aspects of the body's functioning. It can serve as a barrier for pathogens, and it can be a little bit advantageous for other metabolic functions that it does. We're going to focus a little bit more on the inflammation in just a moment. But first, I wanted to look at medical conditions that have been associated with changes in the gut microbiome. In some cases, it's that immune system signaling. So in some cases, what they'll see is that the composition of the microbiome is slightly different than other individuals who don't have a particular condition. Allergies, for example. So if an individual has quite a few different food allergies or environmental allergies, they're finding that the makeup of their gut microbiome looks a little different than someone else who doesn't have those allergies. In some cases, it may be that those organisms are a little bit more sensitive. In other cases, maybe that changes because, well, if I'm allergic to dairy products, then that's not something that's going to be in my diet, and maybe that changes and shifts what the composition of my microbiome looks like. But there are quite a few where they have been looking at this association, and you can see them listed here. We're going to look at inflammation in particular, and related to inflammation, we're going to focus on irritable bowel disease, so Crohn's disease and ulcerative colitis falling under that umbrella, as well as obesity. So both of those families of conditions have been associated with ongoing inflammation, and we'll look at what information we have on the gut microbiome's role in this inflammation. And that is not an all-inclusive list, but that was some of the more common ones that I ran across in a few of the literature resources. So let's focus just on the microbiome and inflammation now. There are a couple components that came up repeatedly that are believed to play a pretty significant role in how the gut microbiome is influencing inflammation in the human body. So one of them are the lipopolysaccharides. So these are large lipid and saccharide molecules that tend to be found in the membrane of gram-negative bacteria. So it's a long chain that tends to be sticking on the outside of some of these microorganisms. Now, in some cases, that's not going to do anything. It helps with signaling. It may help in some cases with attachment to different areas. But those LPS, those lipopolysaccharides, can serve as an indicator. They can serve as a messenger to subsequently turn on different cells within the immune system. I'm going to use macrophages, for example. They may turn on some of the macrophages, resulting in release of a number of different cytokines. Specifically, they tend to be pro-inflammatory cytokines, and we're going to look at that in just a second. On the flip side, part of the microorganisms' breakdown of the food that they are exposed to, it results in the formation of short chain fatty acids. These short chain fatty acids have been identified, 3 of them in particular: butyrate, propionate, as well as acetate. These 3 compounds seem to be beneficial. They help with signaling and seem to turn on other areas of the immune system, including different macrophages, to release different cytokines that tend to be anti-inflammatory. So looking at this in a slightly different way, because that was a lot of words, we're going to focus on the macrophage role, because that's been investigated pretty closely, and it actually nicely looks at both the lipopolysaccharides as well as those short chain fatty acids. So for macrophages, there are classically activated macrophages, those that are activated by pathogens. These could be stimulated by interferon gamma, as well as those lipopolysaccharides, so those long chains that are associated with the gram-negative bacteria. When these macrophages are exposed to these compounds, we end up with the release of a number of different pro-inflammatory mediators. TNF-alpha has been associated with a number of the different interleukins. On the other side, there are different macrophages that have been classified currently as alternatively activated. So they're not as activated by interferon gamma or the lipopolysaccharides. They're stimulated by interleukin 4, potentially interleukin 13. They also may have some signaling come from the short chain fatty acids, and it results in decreases in some of those pro-inflammatory compounds. So we see anti-inflammatory effects. So the natural killer compound may be decreased somewhat. In some cases, we see lower concentrations of things like TNF-alpha or some of the other interleukins that tend to be more pro-inflammatory. So one example of how signaling, because of the gut microbiome compounds like the lipopolysaccharides and those short chain fatty acids, can lead either to an increase in inflammation or potentially to a decrease. So how does this actually look? So it gets quickly into the details of a number of compounds. So I wanted to take a step back. The first of the 2 examples we're going to look at is obesity. So the gut microbiome and obesity. They've looked at a number of different models, animal models as well as human models. And they have found that there are differences in the makeup of the gut microbiome. So in some cases, they see increases in strains. In other cases, they'll see decreases in some of the bacterial strains. Interestingly, which came first, I'm not here to talk about that, but just what they find is that there is a difference in the composition as well as body mass index in some of those models. In some of the obese patients, they have expanded adipose tissue, thus the obesity. Up to 30% of that adipose tissue may be made up of macrophages. So we just talked about macrophages potentially playing a role that in either direction, being pro-inflammatory or being anti-inflammatory. So one example that they've been testing out, granted this was done in more lab type circumstances, they saw that decreases in some of the bacterial strains. So in this case, we're looking at the bifidobacterium. There was a decreased concentration of that. As a result of there being a decreased concentration, we saw a decreased production of glucagon-like peptide 2, so GLP-2. GLP-2 plays a pretty significant role in intestinal epithelial permeability. It plays a role in intestinal proliferation. It inhibits intestinal epithelial apoptosis. It enhances barrier function, as well as assists with digestion, absorption, and blood flow. So by breaking down this compound that helps maintain the intestinal barrier, we get increased epithelial permeability. With that permeability, it means that those lipopolysaccharides, so those long compounds that are associated with the gram-negative bacteria, can cross. It can cross into the small intestine. It essentially can cross into different areas of the body, including the bloodstream, where it didn't usually have access. By having some of that access, what then sometimes happens is there is an increase in the number of macrophages that are making up the adipose tissue. So it signals that more of the macrophages should come to the adipose tissue, and we see a growing of the contribution of macrophages to that adipose tissue. The other thing that occurs is this may turn on a signaling cascade, which results in those alternatively activated macrophages, the ones that were anti-inflammatory, being converted to those traditionally activated macrophages that are pro-inflammatory. So we see more macrophages showing up, and then we see a shift away from the anti-inflammatory to the pro-inflammatory mediators. So we're seeing way more of the production of some of those cytokines, including things like TNF-alpha. The other thing that happens along with this process is there are, maybe because of the concentrations of some of those bacterial strains, we see decreases in those short chain fatty acids. So butyrate, for example, tends to really help bolster the anti-inflammatory signaling. Well, if there aren't a lot of those bacteria there to create the short chain fatty acids of butyrate, then we don't see quite as many of them. And that may essentially further open the floodgates to allow more macrophages into the adipose tissue and allow more of that conversion from the alternatively activated anti-inflammatory macrophages to the pro-inflammatory macrophages. So playing a role, are they what caused the obesity initially? No, we don't have that information here, but these are just a couple models that were looked at in 3 or 4 different ways to be able to see, well, what exactly is going on here with this different distribution and how may they be connected? So this is one story. The other story I wanted to look at just briefly is the gut microbiome and the inflammatory bowel diseases, so Crohn's disease as well as ulcerative colitis. So because of this condition, there can be disruption in the intestinal microbiome. So maybe because an individual is under stress, maybe because they're ill, because they're dehydrated, maybe their diet is a little bit different. There can be a number of internal and external factors that start to make some changes to that intestinal microbiome. They've done a number of studies in both human and animal models that do see that we see a different distribution than what we see compared to individuals who do not have inflammatory bowel diseases. So with some of the changes in those bacterial prevalence, how much we're seeing of some of these, we again can see a shift in those short chain fatty acids. With that shift in the short chain fatty acids, sometimes that includes being anti-inflammatory and protecting some of the other microorganisms that can help with intestinal permeability. So we see more pro-inflammatory compounds. We may start to see a breakdown of the intestinal barrier and see much more permeability, again, allowing things like the lipopolysaccharides to have a greater diffusion instead of just staying in the lumen while we want it where they should be. As a result of that, we can see a shifting of those chemical mediators. So in these particular models, they didn't see as much of a shift from those alternatively activated anti-inflammatory macrophages, they actually saw the development of a different subset of macrophages. So in the intestine, there are a handful of macrophages. There's a whole colony of them that don't respond to those lipopolysaccharides. They don't have the receptor for it. They're called CD14 negative. So they're not going to respond to the lipopolysaccharide to be like, oh, hey, we need to do pro-inflammatory mediators. But in patients who have irritable bowel disease, they see a unique subset that shows up that actually is responsive to those lipopolysaccharides, and it results in an increase in some of the pro-inflammatory mediators, things like interleukin 23, as well as TNF-alpha. So again, not necessarily saying that these shifts caused the inflammatory bowel disease, but these are some indicators, some findings that they've looked at both in animal as well as in human models, that we see more of these compounds. Here may be one of the sources of the pro-inflammatory cytokines that are really helping to continue to fuel this disease process. So in summary, this was just scratching the surface. This is a very, very big area. It was fascinating to get a chance to look into. But the gut microbiome is a unique compilation of microorganisms. It's unique for most different individuals, and it can be influenced by a number of different internal and external factors. The role of the microbiome for several medical conditions is being investigated. It appears to be correlated with the number of these medical conditions, not necessarily the cause just yet, and inflammation is one of them. The microbiome does seem to be able to play a role in amping up the inflammatory response or in dampening the inflammatory response by rebalancing and creating a lot more of those anti-inflammatory mediators. So for the first look at our gut microbiome, I really hope this was helpful, and thank you for your time.